Hey guys, this video is going to talk about the structure of DNA. One thing we talked about in class was the sugar phosphate backbone with the nitrogen base rung. We look at DNA like a spiral ladder, but it's actually called a double helix, and that's important to note. Spiral staircase, twisted ladder. This is our genetic code. Okay, made up of, we'll talk a little bit about, in this section, we'll talk about DNA structure, we'll break down those nitrogen bases, we'll talk about which ones are complementary, and then we'll look at some triplet codons and the production of amino acids in producing proteins. Okay, moving on to the next slide. This twisted ladder can be separated into what we call the sugar phosphate backbone. And you'll see here we have a sugar and we have a phosphate. So this here is the phosphate, I'll just put a P, and that's our sugar. Now notice that the nitrogen base actually attaches to, trying to find a color that doesn't look like that. So the nitrogen base actually attaches to the sugar here, okay? So see that sugar, that nitrogen base, it attaches to this sugar. And then there's a phosphate, and then there again would be a sugar, and then there would be a phosphate, and then on every sugar would be a nitrogen base. Okay, if that makes sense. All right, so these DNA nucleotides, the phosphate sugar backbone is going to be on each side of the DNA molecule, okay, or DNA. So if I was to draw this, for example, okay, it's going to look like, if we unwound it, like a ladder, okay? But what's different about that is on the side, I'm going to have sugars here, okay? On each of these are going to be sugars, okay? And then in between those would be phosphate in blue here. So it would be a phosphate there, and a phosphate there, and a phosphate there. Now on your diploma you'll probably see these diagrams, like the sugar might be a hexagon and the phosphate would be like a square, okay, showing that backbone. And you need to know that these nitrogen bases that I'm about to show you, these nitrogen bases here, there's going to be two of them separated by a line in the middle, but these nitrogen bases here are going to attach to the sugars. Okay, so there are 4 nitrogen bases. These are in your data booklet if you want to find them. A, C, T, G, so adenine, cytosine, thymine, or thymine, and guanine. Okay, so now here's the thing though. A and T are complementary. They will always pair up. Okay, C and G are complementary and always pair up. So it could be T, A, T, doesn't matter. C, G, G, C, doesn't matter. But this one, and I don't know why they don't ever show them together, but that one and that one will pair up, and this one over there, and that one will pair up. Okay, so if you don't, they will never pair up or will get a mutation. Okay, so moving on to the next slide, so you'll really get a good glimpse of what I'm talking about here. So the sugar and the nucleotides are attached. In between is a phosphate holding the sugars together here, and so sugar, phosphate, sugar, phosphate, sugar, phosphate, so on and so forth. And then nitrogen bases in the middle. T and A pair up here. G and C pair up. A, T, C, G, and that makes our code. But these are called base pairs. Now, and they're complementary. So now, if I was to separate this down the middle and take a look at this strand, each three nitrogen base section is going to make a triplet codon. And triplet codes, which are going to code for amino acid, and a series of those are going to make proteins. So I'll talk to you about that on the next slide, but you must know that sugar phosphate backbone. You must know the nitrogen bases, and you must know which ones are complementary. As if I was going to write a strand out for you, you should know which one is which. Now, the next thing I'm going to talk about first is DNA replication. So in DNA replication, basically what happens is the DNA makes an exact copy of itself. Okay, so it makes an identical DNA molecule. And so what's going to happen is it's going to unzip. So you need to know these 3 steps here. So step number 1 is going to have the DNA untwist, and it's going to unzip, and it's going to break those bonds between the nitrogen bases, and you can see that happening here. So now what happens is once these bonds are broke, like in this section here, then what's going to happen is floating nucleotides are going to attach to the exposed halves. Now they're going to attach to their complementary strands. And then essentially what you're going to get is that you're going to see here a strand and then an identical strand here. Okay? So this is DNA replication. So that's going to occur. So understanding how that works. Still notice that this right here is a sugar, and these little bits here are a phosphate, sugar phosphate backbone on both sides, nitrogen bases in the middle, A, T pair together and are complementary, and C and G are complementary. And so again, moving on, now we're going to talk about triplet codons. So we've got the structure of the DNA, and we understand that. But now we need to talk about how proteins are produced. And remember, proteins are going to build and repair within our body. They have several different roles in our body, but we're going to talk about a few. So basically what's going to happen here is we are going to see an amino acid plus an amino acid plus potentially more amino acids is going to eventually produce a peptide or a protein, but basically we're building proteins. So we use our data booklet for this, and everyone should have a copy of the data booklet. But there's 20 possible amino acids, and basically they join in different combinations to make a variety of different proteins. So amino acid plus amino acid plus amino acid, blah, 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 and then whatever the sequence is going to produce different proteins. But how do we start the production? Well, there is one starting protein that's going to start and begin reading those codons and basically producing those proteins in that sequence, but it has to start producing the protein. There are also 3 that act to stop a protein production, and that means that that protein is created. Okay, and so basically what I can show you here is I can show you the chart from your data booklet so you can get an idea of what this actually looks like. So I'll zoom in on it for you a little bit after, but basically what happens here is if I was to give you this code, so say I gave you, I just have to think, I should have had this prepared. Let's say I give you this code, okay, and then I give you this here, T, T, A, and then I give you A, C, and then I give you TGA. Okay, so what we're going to want to do to understand this chart is we're going to want to split these up in 3 starting left to right. So I'm going to go, there's 3, there's 3, there's 3, there's 3. Now, this one, ATG, I take a look on my chart here, and I'm looking around. There's actually a way where you can use these letters to find it, okay? But I realize that it is right here. So ATG is my start codon. Now the next one is TTA. So now I have to find TTA. So I recognize it right there. and I see that it is leucine. Okay, and then next one is AAC, and what I'm going to do is I'm going to locate this, and I take a look here. Okay, and it's asparagine. Okay, and I see that, and these are amino acids. Okay, and then I see TGA, and I look here and I go, oh, it's a stop protein. So this, all those amino acids will produce a protein, name unknown. But now this is different than what you are used to with RNA and Bio 30. We don't do that in the Science 30, so this is what would be expected on your diploma. So that's how you use this chart. It's on page 13 of your data booklet. So hopefully we're understanding the structure the structure of DNA. But the whole purpose of this is to produce proteins. So I'll just talk a little bit about some of these proteins that we actually discuss. So enzymes speed up chemical reactions. Structural give us support and framework. Basically, they allow us to attach different muscles, and those proteins just help us build and give us that support we need. Transport, if we're going to move things within the body. Hormones act as signals, and they coordinate the body as well through those signals. Contractile proteins, they just change shape, and they can allow for large movements. Defensive proteins, they basically protect the body against disease. And then energy proteins just provide us with that chemical potential energy we need in our body. Okay, so there's some examples on the right. I'll let you take a look at those. But you will need to know a few of these proteins for your quiz and your exams and such. And you'll have to know some of the roles of proteins on the diploma possibly as well. Okay, so I'm going to move. Actually, I'm just going to end it there. So these proteins on structure and function, or the structure of DNA. Okay, so phosphate, sugar backbone, nitrogen bases, A and T are complementary, C and G are complementary. DNA replication is the unzipping, and then floating nucleotides will reattach to their complementary nitrogen base and produce two identical strands of DNA. And proteins, there's a variety of proteins that are used for the structure and providing support and the building of our body, but also on the repair end as well. And some of them send signals, but they're all really important. So we use the triplet codons that produce amino acids, and a series of those amino acids will make a protein. So anyways, hopefully that's helpful. I'll see you in the next video.